So today we're going to talk about amino acids. Amino acids are the building blocks of protein. Protein acts as enzymes, hormones, receptors, channels, transporters, antibodies, and support structures inside and outside of the cell. So right now we'll look at the structure of an amino acid. So the amino acid will have your alpha amino group, your tetrahedral alpha carbon, and your carboxylic acid. So H2N attached to your tetrahedral carbon, and then this is going to be attached to your carboxylic acid. And then we have your R group, and then a hydrogen attached to it. And the R group is going to vary for every amino acid, and this is going to make each amino acid unique. And the alpha carbon is going to be attached to 4 different groups, except for glycine, and this carbon right here is going to be a chiral center. A chiral center means chirality, and this means that a molecule that has optical activity and can rotate in a plane. So there's going to be 20 amino acids, and each amino acid is going to have the same backbone, which is going to be nitrogen, carbon, carbon. And a unique feature in its side chain in the R group gives its physical and chemical properties. So let's look at the hydrophobic nonpolar amino acids. These are going to have aliphatic side chains and aromatic side chains. These hydrophobic residues associate with each other rather than water, so they dislike water. And these are going to be found on the interior of folded globular proteins. So the hydrophobic nonpolar with an alkyl group, there's going to be 7 of them, and they're going to consist of glycine, alanine, valine, leucine, isoleucine, methionine, and proline. And there'll be 2 hydrophobic nonpolar that are going to be aromatic. And this is going to be phenylalanine and tryptophan. Now we're going to look at the hydrophilic polar amino acids. These have an R group that is polar enough to form hydrogen bonds with water, but not polar enough to act as an acid or base. Hydrophilic means react with water whenever possible. So the neutral amino acids are going to be tyrosine, serine, threonine, cysteine, glutamine, asparagine. The acidic amino acids are going to be glutamic acid and aspartic acid. And then the basic hydrophilic polar amino acids are lysine, histidine, and arginine. So the hydroxyl groups with an R attached to an OH group, these are hydrophilic polar amino acids, and these are going to be tyrosine, serine, and threonine. And these all have hydroxyl groups that are often modified by attachment of a phosphate group by the regulating enzyme kinase. The structure changes because the phosphate group is very hydrophilic, and this is important for regulating proteins. So the acidic amino acids are glutamic acid and aspartic acid. These are the only amino acids with the carboxylic acid, and their pKa is about 4. The basic amino acids are going to be lysine, arginine, and histidine. Lysine has a pKa of about 10, arginine has a pKa of about 12, Histidine has a pKa of about 6. So histidine is very unique. Histidine is unique because it has a pKa that is very close to physiological pH. At pH 7.4, histidine can be in protonated or deprotonated form. And a way to remember this is his goes both ways. So it can be protonated or deprotonated. And then we have sulfur containing amino acids, which is cysteine. And this contains a thiol, an SH, and is fairly polar. And then we have methionine, which is a thiol ether, and this is nonpolar. And cysteine is going to play an important role in the disulfide bridge that we will talk about in a little bit. So now we'll look at the amino acid proline. This is going to be a secondary alpha amino group. And we'll look at the structure and we'll see why this creates a kink in the secondary structure. So proline is going to look something like this, and this is going to be the only amino acid that has a secondary alpha amino group. So right here. Now we look at glycine. This is also going to be very unique. This is achiral. It does not have a chiral alpha carbon and is an alpha helix breaker and creates kinks in the secondary structure. So if we draw glycine, we have H2N attached to your alpha carbon, and then this is attached to carboxylic acid. But instead of having an R group, it's going to have H, a hydrogen, and another hydrogen attached, making this achiral. Something to note is the essential amino acids. And these are going to be valine, leucine, isoleucine, phenylalanine, tryptophan, methionine, threonine, and lysine. Now we're going to look at the protein structure and how protein is formed between amino acids. So how are amino acids and proteins bonded? Through covalent bonds. And the linkage of amino acids into polypeptide chains is called the peptide bond. And these are formed by alpha amino groups donating electrons to the carboxyl group, and there is a loss of water. So here we can see the alpha amino group. These electrons right here are going right into the carboxyl group. And then as this forms, we're going to lose water. And then if we look here, each of these amino acids, when they connect, they're going to be known as a residue. So as this connects to the other amino acid, each of these are known as residue. And if we have an oligopeptide, something that looks like this, so we have phenylalanine attached to glutamine, attached to glycine, attached to serine, and it's attached to alanine. What number of acid and base amino acids are in this? And the only one is going to be glutamate, which is acidic, and is the only acidic or acidic functional group in the oligopeptide. And then we have phenylalanine, which is going to be the free amino group because it is on the N terminus. So this over here is the N terminus. And then alanine on the end is going to have the free alpha carboxyl group. So now looking at thermodynamics of amino acids, thermodynamics states free energy must decrease to proceed spontaneously, and that such a reaction will spontaneously move toward equilibrium. So at equilibrium, which is thermodynamically favored, dipeptide or amino acids? Well, since the dipeptide has higher free energy, it is less favorable. Existence of the chain is less favorable than existence of isolated amino acids. How are peptide bonds formed and maintained inside cells? Protein synthesis causes stored energy to force peptide bond formation. So once the bond forms, its destruction is thermodynamically favored. It will remain stable because activation energy for hydrolysis reaction is so high. Hydrolysis is thermodynamically favorable but kinetically low. Now we look at hydrolysis of a protein. So there's going to be acid hydrolysis, which is combined with heat and is nonspecific, and then proteolysis or proteolytic cleavage. This is going to have specific cleavage, and this protein that does the cutting is known as protease. So the enzyme that is going to cut the protein will be protease. And many of these enzymes are only going to cleave the peptide bond adjacent to a specific amino acid. So if we have protease trypsin, this typically will cleave on the carboxyl side of positive charged residues arginine and lysine, as chymotrypsin cleaves adjacent to hydrophobic residues such as phenylalanine. So based on the information above, if the following is cleaved by trypsin, what amino acid will be on the new N-terminus? Well, since this cleaves on the carboxyl side, if we look, it'll cleave right here, and then we're going to get two fragments. One of phenylalanine attached to phenylalanine attached to lysine, and then the other fragment is going to be alanine attached to glycine, attached to glutamine, and attached to lysine. Now we'll look at the disulfide bond. So the disulfide bond is created by cysteine, which has a very reactive thiol, SH, in the R group. Thiols react with each other, and if two cysteine are close together, they react forming a disulfide bond. The disulfide bridge plays an important role in stabilizing tertiary proteins. And when in a disulfide bond, the cysteine SYST INE, this is going to be the disulfide bond. So when cysteine turns into cysteine. And a way to remember this is oil Riggs redox reactions. So oxidation is lost and reduction is gained. So disulfide bridges form extracellular or the exterior of the cell because it is in an oxidizing environment. So the disulfide bridge, cysteine, is formed extracellular or in the exterior of the cell. As for the cysteine, these aren't bonded. They're the intracellular or inner cell because there's antioxidants that make it a reducing environment. So again, these are going to have an antioxidants. So it basically is in a reducing environment, and these are going to have their hydrogen atoms. And then when it's oxidized, this loses the hydrogens and then creates the oxidizing environment and attaches the two S atoms. Now we look at the primary structure. This is going to be the amino acid sequence. So linear ordering of amino acid residues, and this is the linear sequence. Amino acid attached to amino acid attached to amino acid. So it's just going to be linear, so like a straight line. And then for secondary, these are hydrogen bonds between backbone groups. This is the initial folding of the polypeptide chains, and they are stabilized by hydrogen bonds between the NH and carboxyl groups. And then there's going to be alpha helix, and then beta pleated sheets. Let me redraw this. So the alpha helix looks something like that, and it has the same dimensions. The alpha carboxyl carbonyl oxygen of one amino acid residue is hydrogen bonded to the alpha amino proton. Proline, glycine, these are going to form kinks in this polypeptide chain and are never an alpha helix. As for the beta pleated sheet, these are stabilized by hydrogen bond between the NH and CO groups. These occur between residues distant from each other. The backbone is extended, and these can be parallel or anti-parallel. So the parallel looks like something like this, and then the anti-parallel will kind of like curve and curve. And the majority of the hormone receptors and ion channels are to be found in alpha helical transmembrane regions, integrated in hydrophobic membranes of the cell. So the alpha helix is going to be very important for finding hormone receptors and ion channels. The alpha helix also favors these hydrophobic transmembranes because all polar NH and CO groups are hydrogen bonded on the inside of the alpha helix and don't interact with hydrophobic membrane in the interior. Now for the tertiary structure, these are hydrophobic and hydrophilic interactions. These are located more distantly from each other in the polypeptide chain. There's many folds that occur, folding of alpha helices from secondary structures into higher order tertiary structures. These are driven by interacting of R groups and water. Hydrophobic R groups fold into the interior as hydrophilic fold out and are exposed to water. So if we draw a globular protein, we'll have our H2 molecules, H2O molecules. These are going to be on the outside. And then our hydrophobic region is going to be right here in the middle. And then your hydrophilic is right here. And this is going to be in the globular protein. So disulfide bonds, these are not great examples for tertiary structure because it is covalently bonded, but they're still considered part of the tertiary folding. Now for the quaternary structure, these are various bonds between separate chains. There's interactions between polypeptide subunits. The subunit is a single polypeptide chain as part of a large complex containing many subunits. And the quaternary structure is going to be the arrangement of subunits in a multi-subunit complex. This has the same forces as secondary and tertiary structures that stabilize its structure. And these are non-covalent bonds, hydrogen van der Waals interactions. Peptide bonds aren't involved in quaternary structures. Now we'll look at the denaturation of proteins. Each protein is folded into three-dimensional structure as required for a protein to function properly. So denatured is the unfolding or improperly folded proteins. This refers to disruption of protein shape without wrecking or breaking peptide bonds. So what can proteins be denatured by? This is going to be urea, extremes of pH, temperature, and changes in salt concentration. And now the last thing that we will look at is going to be the Fischer projection. of amino acids, and this is going to give us an idea of the L and D amino acids. So if we look here, this amino group is going to be on the left, so this is going to make it an L amino acid. As for this one on the right, the NH2 group, the amine group, is going to be on the right, and this makes this a D amino acid. So these are non-superimposable. These cannot be placed on top of one another and give the same molecule. Therefore, these are enantiomers. L-amino acids are constituents of proteins. The body synthesizes most of its L-amino acids, and these get incorporated into proteins.